Good afternoon, dear students. Topic of the lesson is ontogenesis, general patterns of the embryonic period, ontogenesis regulation. Key equations are presented on the slide. Ontogenesis is the individual development of a person. The term was coined by Ernst Haeckel in 1866. Definition of ontogenesis: individual development of an organism. Ontogenesis is the sequential development of each individual, which is based on the implementation of genetic information at all stages of existence under certain environmental conditions. There are two main types of ontogenesis: direct and indirect. Indirect development occurs in the larval form, and direct development occurs in the non-larval and intrauterine forms. Direct, in which the body of the mother or egg shells levels an individual that differs from the adult organism only in smaller size. Birds, mammals. The non-larval type, in which the embryo develops inside the egg, and intrauterine type, in which the embryo develops inside the mother's body and is connected to it through the placenta. With transformation or metamorphosis, in which the larva emerges from the egg, orange, simpler than an adult animal. After all, it has special larval organs that are absent from an adult animal, and is not capable of reproduction. Ontogenesis has three main periods: pre- and embryonic and post-embryonic. Pre-embryonic, remember, progenesis. The embryonic period is divided into stages of cleavage, gastrulation, histogenesis, and oogenesis. The post-embryonic period is divided into pre-productive, reproductive, and post-productive periods. Remember, progenesis includes gametogenesis and fertilization. The process of germ cell formation is called gametogenesis. The formation of the good oogenesis occurs in the female gonads, the ovaries. The process of sperm formation is spermatogenesis in the testes. Fertilization is the process of combining male and female gametes, which leads to the formation of a zygote and the subsequent development of a new organism. During fertilization, two processes take place: egg activation and syngamy. Cleavage is a series of successive mitotic divisions of zygote and blastomeres, ending in the formation of a multicellular embryoblastula. The first fragmentation begins after combining the hereditary material of the pronuclear or synchorion and forming a common metaphase plate. The resulting cells are called blastomers. The peculiarity of mitotic divisions of cells is that with each division the cells become small and smaller until they reach the usual ratio of the volume of the nucleus and cytoplasm for somatic cell. Between the next divisions there is no cell growth, but the DNA is necessarily doubled. As a result, mitotic cycles shortened and fragmentation is rapid. First, the blastomers adjacent to each other, forming a cluster of cells moreover. Then cavity is formed between the cells, blasted shell filled with fluid. The cell pass to the periphery, forming the blastula or the blastoderm. The total size of the embryo does not exceed the size of the zygote. The result of crushing is the formation of a single layer embryoblastula. The pattern of cleavage is greatly influenced by the presence of yolk. It can be holoblastic, symmetrical and asymmetrical, and meroblastic, discoidal and superficial. The structure of the blastula will depend on the type of crushing. With the holoblastic type of crushing, a typical blastula is formed, a holoblastula. With a complement uneven type of crushing, and amphiblastula is formed with micromers at the animalic pole and macromers at the vegetative pole. The blasted cell is displaced to the animal pole. In plant-cell mammals, including humans, a blastocyst is formed, consisting of the embryoblast, from which the tissue of the embryo and trophoblast cell of the extra-embryonic part will develop. Gastrulation is the process of formation of a multi-layered embryo. In the process of gastrulation there are two stages: formation of ectoderm and endoderm, and formation of mesoderm. There are four main ways of ectoderm and endoderm formation: invagination, epiboly, delamination, immigration. Invagination, the hole through which the formed cavity communicates with the external environment is called and blastopore. Epiboly are growths of small cells of the animal pole or large cells of the vegetative pole, absurd and amphibious. And delamination, Stratification of blastoderm cell into two layers, living on top of each other. It is observed in reptiles, birds, egg-laying mammals, and in the embryoblast of placental mammals. Immigration, movement of groups or individual cells into a blasted cell. There are also 5 methods mixed, combining different ways of forming two germ layers. In placental mammals, germinate and immigration. The formation of tissue and organs, histo and organogenesis. Phase of organogenesis, one neurulation, and the phase of building of the bodies, the formation of internal and external organization feuds. Neurulation, the formation of a complex of axial organs. The embryonic stage of neurulation is called a neurula. On the dorsal side of the neural plate is formed its edges, chicken and rise, forming a nervous cushions. In the center of the plate there is Y-shaped depressions, a nerve groove, its edge touch, and the close. As a result of this process is a neural tube with the cavity of neuroculm. From the ectoderm material, in addition to the tube, the epidermis and its derivatives, hair, nails, claws, skin glands, components of the organs of vision, hearing, smell, oral epithelium, and animal develop. Mesoderm is separated into dorsal and ventral regions. Dorsal segmented and presented by somites. The ventral part is called cytolamina. Somites are connected with the cytolamina by the intermediate mesoderm. From the mesoderm develop all types of connective tissue, dermis, skeleton, striated, and small nucleus, circulation and lymphatic system, sexual system. From the endoderm develops the epithelium of the stomach and intestinous liver cells, secreting cells of the pancreas, intestinal and gastric glands. Embryonic induction means that one tissue and groups of cells can influence the development of other cells. Embryonic induction. Embryonic induction means that one tissue or group of cells can have influence on development of other cells. Such phenomenon was discovered by Spearman and Mangold. Some provisional organs are made from embryo material. The most primitive is the yolk sac. It first appears in fish embryos. It has endodermal region. It is a membrane with many vessels, which surrounds yolk storage. It serves for transmitting nutrition from yolk to an embryo. It is great importance for reptilians and birds because their eggs contain much yolk. The mammalian embryo also forms the yolk sac, but it has no function for embryo. New three professional organs were formed for defense and embryo nutrition in reptilian, bird, and mammalian embryo. They are amnion and allantois. Chorion is developed from the external layer of the primary body fold. In reptilian and bird sex, the chorion touches the eggshell, whereas in mammalian embryo it touches uterine mucosa. It forms externally incorporated into outer wall. This also it helps to excrete waste products of the embryo. It grows together with an embryo to provide sufficient supply for it. Amnion is developed from the internal layer of the primary body fold. The space between the amnion and embryo is called the amniotic cavity. It is filled with fluid secreted by the amnion and embryo. Amniotic fluid prevents embryo water loss, serves as defense pillow, and provides conditions for embryo movements. With the chorion with uterine tissue from the placenta. The placenta provides water and food supply uh, for the embryo. Also, it helps to excrete waste products of the embryo. Allantois is a diverticulum of intestine. It grows between amnion and chorion. In reptilians and birds, the allantois serves as a place of nitrogen waste products storage. The allantois use with the chorion making and allantois chorion membrane which is rich in vessels. The yolk sac has no specific function in a human embryo. The human have a small allantois. It contains vessels coming to the placenta. The human prenatal development has three periods: primary, embryonic, and fetal periods. Intrauterine development lasts 13–14 weeks. Link to book 1 slide. Ontogenesis is regulated at several levels. gene, epigenetic, cellular, fibrillary organ, organismic, biocyanotic. Critical periods of ontogenesis. The study of animal development resulted in discovery of the so-called critical periods of embryogenesis. The term is used to point the period when the embryo is very sensitive to various harmful influences which can result in developmental defects. Organisms' sensitivity varies in different embryogenesis stages. In some periods, the embryo is more sensitive to chemical, in others, and an embryo is more sensitive to temperature change. Critical periods are characterized by the increased metabolism and respiration and decreased of growth rate. There are critical periods in development: gametogenesis, fertilization, implantation, development of actual mucus and placenta, increased growth of the brain, formation of organs, differentiation of the sexual apparatus, birth, newborn period, puberty, menopause. References. Thank you for attention.